What does life truly need to exist? To answer that, scientists have been searching for the minimal genome, the smallest number of genes an organism needs to survive and reproduce. This search helps us understand the bare essentials of life. It also fuels progress in synthetic biology, where researchers aim to design artificial organisms that could be used in medicine, industry, and beyond. Discovered in 1981, Mycoplasma genitalium is one of the simplest life forms ever found, with just 525 genes compared to E. coli's 4,000. It survives by relying on its human host for nutrients. Its genome became so small through evolutionary pressure in the nutrient-rich but highly competitive environment of the human genital tract. Despite its simplicity, these 525 genes still cover the essentials. Around 15% are involved in copying DNA, 25% handle protein production, and 30% support basic metabolism. The remaining genes help maintain the cell structure and manage interactions with the host. Even this ultra-minimal organism operates close to the absolute limits of what life needs to function. In 2016, scientists built a synthetic organism called JCVI-SYN 3.0, containing just 473 genes, even fewer than what we see in nature. It was a huge breakthrough, but it came with trade-offs. This minimal cell grows 3 times slower than its natural version and often divides in strange, uneven ways. Extreme simplification has its costs. What's even more surprising is that 149 of those 473 genes had unknown functions. That means nearly a third of genes essential for life are still a mystery to us. Today, researchers estimate that the true minimum for life might lie somewhere between 250 and 400 genes. But going that low creates serious problems. Without genes to repair DNA, mutations build up and can be deadly. And if you strip away too many metabolic functions, the organism becomes completely dependent on a host to survive. The lines between life and non-life start to blur when we look at things that aren't even cells. Viruses, for example, can have as few as 10 genes, but they can't do anything on their own. They rely completely on a host to reproduce. Then there are viroids, which are just tiny loops of RNA, and prions, which are misfolded proteins that somehow spread by making other proteins misfold too. These strange entities challenge our usual ideas about life. They suggest that maybe it's not about having a cell or being independent. Maybe the real core of life is just replication and information transfer. Could life exist with just 100 genes? Surprisingly, the answer might be yes. Some scientists imagine early forms of life made of self-replicating RNA, able to copy themselves without any DNA. Others propose chemical systems that keep themselves going through simple feedback loops. But there's a big catch. With so few genes, these tiny life forms would be extremely fragile. They could easily fall apart under the weight of random mutations, something called an error catastrophe. Too many mistakes, and the organisms stops working together. The quest to define minimal life raises profound questions. Is life simply self-replicating chemistry, or must it also have independence? If something can't survive without external support, can we truly call it alive? As synthetic biology progresses, we may be forced to rethink and redefine what life really means. Mycoplasma, Genitalium, and JCVI scene demonstrate that life persists with hundreds of genes, but the true minimum remains elusive. The search for minimal genome is more than scientific curiosity. It's a fundamental exploration of what it means to be alive. Whether life can survive with just 200, 100, or even 50 genes is an open question, and the answer could redefine what we consider the minimum requirements for life, reshaping our entire understanding of biology. Thanks for watching. If you found this video helpful, give it a thumbs up and don't forget to subscribe to the channel.